So you've seen that slide, I think, before. It illustrates different shapes of dendritic trees, and we know that these neurons evolve these different shapes clearly to have a particular wiring of the brain, a particular connectivity that is enabled by it. So for example, the Purkinje cell, dendritic tree, is flat and is very, very dense, and it's therefore able to read out potentially every parallel fiber, every input axon that crosses it. And the Purkinje cell can choose among those 100,000 or so inputs which ones to pay particular attention to. Whereas the pyramidal neuron here on the right, for example, is very sparse and samples mostly down where the soma is near layer 5 and up in layer 1. And we will also be able to study the meaning of that. But in addition to the different connectivity, each of those different geometrical structures of the cells has direct consequences for how those neurons treat their inputs. How strong, for example, the influence is of an input at layer 1 at the soma of a layer 5 pyramidal cell. And it's not the same as an input in layer 5. It's an undemocratic neuron. Whereas the Purkinje cell is far more democratic. All these inputs have about the same power of influencing the soma. And there's more complex interactions that are possible at the level of the different inputs on the dendrites. So the question we're interested in is, how do the neurons treat the different inputs to create an output spike? What's the algorithm that should replace the big question mark here? And here's an alternative way of thinking about this question, which is, what are the functional compartments in neurons? Is it a single spine? Is it a cluster of spines? Do they interact locally? Is it a whole branch or a whole subtree? And the domain at which we want to describe this, you've heard now several times this morning and afternoon, is the domain of describing dendrites as electrical cables. They have a certain axial current that flows along the cable, and we can measure that in kind of imagined cross-section. And there's a membrane current, capacitive and ionic, that goes through the thick lines, the membranes. And we have the cable equation describing that, and the variables that we want to solve for are the voltage at each point, the difference between inside and outside, the axial current, and the membrane current. And the parameters are these. So before we can get any numerical results for the voltage as a function of time and the axial current and the membrane current, we have to choose particular parameters and have to choose wisely. One way of choosing wisely is to fit experimental data, as just being explained by Dieter. And so these are typical values for the passive parameters that you get. So the intracellular resistivity is on the order of 70 to 150 ohm times centimeter. Membrane capacity is 1 microfarad per centimeter square. And so these are not very variable, but between cells the membrane resistance can vary a lot because the amount of leak channels and other channels that contribute to it varies a lot between cell types. And that's the one end of the problem domain neuron is addressing, and the other end is entire networks of maybe simpler spiking neurons in, for example, the spirit of this Brunel network, a famous network that I think you will hear about in the next few days. For example, by Sasha van Albada. So, what kind of parts does neuron exhibit? First, there's the back end. There's the actual numerical engine that solves the cable equation efficiently. And you take advantage of all the tricks and design ideas of Michael Hines and others that contributed to doing good and efficient numerics on this problem. Then you need a way of describing your problem, right? So which parameters exactly are we using? Well, there has to be a language for describing which membrane resistance is where and which membrane capacitance is where and how thick the dendrites are and how they are connected. And that language in the old days was called HOC. You're going to teach this. But in the modern days, there is a way of implementing this in Python, if you like Python and want to use the entire universe of Python code to create and then analyze your simulations. That's something you can do. So these are interpreted, but it's okay because it's like a high-level description. Morphology is only read once, and then the simulation is run with that. And so that doesn't have to be compiled. But things like the sodium channels and the synapses, they are compiled. And so we have something called N-model, neuron model description language, that allows you to describe a Hodgkin-Huxley model or a synaptic current. And that's compiled and then linked with the rest of neurons, so it's fast. We have a graphical user interface. You're going to experience it in the workshop. It's kind of old school. It's a bit complicated to use, but it's great for prototyping. It's great for playing around and also for teaching. Once you have figured out a problem you want to work on and figured out a way of solving it, of programming it, you will less and less use the graphical user interface. You will have your scripts that are running reproducibly the simulation multiple times, and then you'll have Python code or other code that analyzes the simulation results outside of the interactive graphical user interface. And finally, we want this to be interoperable with Python, the whole universe of all the solutions that people in the whole world have implemented in Python. You want to be able to share data in a common format, so Maria-Lena talked about that. We want to have exchange between different sources of morphologies, for example, all of this. So it has to speak to the rest of the world. So these are the ingredients. We have a way of formalizing neuronal morphology. On the top left, you see a stylized neuron with the big bulge being the cell body and dendrites going to the right and branching off. And the axon, starting with the axon hillock, AH, and then internodes, which are myelinated, and nodes of Roth, which are called N, going down towards the target neurons. And in the neuron simulator, we are dividing this into so-called sections. And a section is an unbranched piece of cable. It may not be an actual really kind of long cable. The soma is also a cable for this instance. It's just fat and short. There's branch points in between where two of those sections might meet, but it can also be more than two. It can be 3 or 4. And the sections then are further subdivided into segments. And so that's a neat way for kind of a biologically intuitive and easy way of separating the actual description of the biological morphology from the details of the numerical implementation of how accurately you want to simulate this, how many compartments you want to donate to every section. The soma might be done with one compartment because it's so short, but a long extended cable might need 10 compartments, but we can keep this flexible. We can separate those. And that's one of the advantages of NEURON, for example, compared to Genesis, where those things were always linked. Genesis, by the way, is basically the competitor to NEURON. It's an alternative way of doing the same thing. There's nothing wrong with it. It's great to have two ways of doing the same thing. Competition, is good, and it allows you to cross-check things and so on. So sections and segments. We'll talk about these in detail also this afternoon. Each segment basically is a replacement electrical circuit that is made of 2 halves of the axial resistance. So these segments are always symmetric in NEURON. The membrane resistance and capacitance is always in the middle of the segment. We don't have asymmetric compartments. For these, you have to use Genesis. And let's say this is how it looks like if you've got 2 segments per section. So basically, you have 1 half axial resistor on the left, you can 2 halves in the middle that are joined up, and another half axial resistor per segment on the right. And you've got, of course, 2 membrane capacitors and 2 membrane resistors. And now we have a certain nomenclature of reading out voltages and addressing these 2 segments. And it works like this. First, the language statement that we want 2 of them instead of the default of 1 is called nseg equals 2. And we'll be using that in a second. And then, if we want to read out the voltage, membrane potential is called lowercase v. We now have 4 possible locations along these 2 segments where we can do it, where there's a valid voltage to read out. And that is wherever you are kind of at the end of a half axial resistor. So there is a voltage at a relative coordinate 0. We have defined a relative coordinate from 0 to 1, going from left to right for the whole segment. And then there is a valid voltage at the middle of the left segment, which is relative coordinate 0.25, and there is a valid voltage at 0.75, and again at 1. So there's no valid voltage at the middle at 0.5. For that to be possible, you need an odd number of segments, then you are fine. Like 1 is okay, or in this case, 1001. So that's why an odd number of segments is always a good idea. So here's our simple model, and it's an unbranched cable. It creates the data structure by using the create statement, and we can choose whatever we want to call this thing. So we call it cable. And then the access statement says that all of the segment commands refer to this section and not any other. For example, we give it a length of 10,000 microns, so a really long cable. We make it thin, and we give it an odd number of segments. And then the only thing we do is to insert a passive membrane. So we make there a passive membrane resistance. Capacitance and axial resistance is already given by default. We use the default values here. You want to be careful that you, of course, set them properly. Here we don't. And so we give this a membrane resistance that's compatible with what a pyramidal neuron would have and a resting potential of minus 65 millivolts. So I've prepared here 2 files, and this is how I run NEURON. And I have to drag this over. So there's a main menu. Here it comes. And there's a run control menu. Comes here. And there's various windows that allow me to graphically modify the simulation. And here's a result. Window. So I'm going to run this simulation by pressing init and run. Boom. And that was fast. So I placed a current injection at the middle of this cable, where the blue dot is, and I've turned on current injection. And what it does is it depolarizes that location, and there's the exponential drop-off that you expect in the steady-state case of the voltage with distance. And now I can use the interactivity of NEURON. For example, I can show the parameters, and I can change the amplitude of this current. These are the kinds of things you will do in the afternoon. So the important thing is to type the new number and then hit return. Otherwise, it doesn't accept the change. Now it's got the check. Now it's got the acceptance. And if I run it again, what should happen? Well, this whole thing should be higher. But the space constant doesn't change. It's linear, right? We have 50% more current. We get approximately 50% more depolarization. Yeah, I can change the duration of this thing and so on. I can also make it slow. So this is very fast. I can artificially slow down the simulation by making a smaller time step. Maybe we can then watch it being depolarized. Yeah, you can almost see it as it goes up. Okay, simple NEURON simulation. I'm going to quit this. To get out, you say Control D for disconnect. So how do I get the PowerPoint back? Oh, I think I pressed something wrong. No. This. Oh yes, I'm good. I did it. Super. Okay, so here are typical variables that we have to deal with. There's a global time in the simulation. It's called t. There's a time step. We just changed that to make the simulation artificially run slow. It's dt, both in milliseconds. Then there's geometry things like length and diameter. The capitalization does matter. So capital L it is and lowercase diam, and they're all in micron by convention. NSEG has no dimensions, just counting how many segments you have per section. CM lowercase is the membrane capacitance. RA, capital RA, is the axial resistivity, intracellular resistivity. And if we have the mechanism pass inserted, we have these things called G underscore pass and E underscore pass, which are membrane conductance. So you can think of it as conductance per square centimeter or 1 over resistance times square centimeter. This is more intuitive, right? So how many channels in a certain little piece of membrane for a certain area. And so this is a built-in mechanism that we don't have to supply, which is the ohmic membrane, and it's called pass. But NEURON is extensible, so it's these N-model files, the mod files, that you can write yourself or take from a library, where you can define your voltage-dependent sodium channel, for example, and it might be called your NA or something. And that would be then the extension. So you have a variable name, underscore, and then the mechanism name, which automatically means that if you define your normal variables that you want to use, like for a loop or for anything you want to calculate, it's better not to use underscore in it. So your variable name should reserve the underscore for things that are part of such a N-model defined mechanism. That's good style. You don't have to do it, but it's better like this. And there's some other global variables which do matter and can trip people up. So for example, sodium channel kinetics depends on temperature. And that's implemented even in the built-in sodium channel, the Hodgkin-Huxley sodium channel. There's a variable called Celsius, and the default is 6.3 degrees, which is this squid axon experiment from Hodgkin-Huxley. Our brain has a temperature of like 37 degrees, and so it's important to set this variable correctly if you use other people's mechanisms. I now want to show some resources that I've found valuable in the line of our work, and the first of them is the NEURON book. So there is, of course, online documentation, and there's downloadable articles that document, introduce, and explain NEURON. But here's the comprehensive presentation of everything you ever want to know in book form. It's very convenient. Next, of course, and I'm really going to show this later then, if there's time left, there is online documentation, which is very good. For example, there's an alphabetical list of commands. You often remember the command, but not the details of it. So you go in the index, you search, for example, for create, and then you click on the explanation. It's very useful. Finally, there is a whole database of ready-to-run published models. So NEURON, one of the big advantages of having a simulator like NEURON is that people can have a standard language in which to describe their models that they can share across the whole world, across labs. If everyone wrote their own simulator, everyone would be an island and would be stuck with their simulator and never able to share and improve upon other people's models. Here, we have something that's curated, where we know things run, and they should behave as in the published paper, if possible. You can even, make a list of your errors in your paper in the README here, as these people did. And then you have a starting point for your own project. You can choose among hundreds of published models, where there's a paper describing the entire background and application and whatever they found with it. And you have the ready-to-run code to reproduce parts of, or maybe the entire paper. And the art is to pick one that's close to what you want to do, so you have an easy way of modifying it towards your purposes. And if you want to, for example, start with a new cell, here's a library of morphologies that also Dieter has mentioned, and that gives hundreds of thousands of neurons to choose from that people have reconstructed by hand and or automatically nowadays. So we're going to browse this if we have time left later. Now I want to tell you about kind of our use case, and really think of it as continuing exactly the path that Dieter has set. So here is a model of a single neuron whose morphology you can see on the left, and whose membrane potential is coded in color on the morphologies. At the moment it's mostly blue, which means resting potential. And there's little circles kind of springing about, and these are the currently active synapses. So this model has 5,001 synapses placed on it, and we activate them using spike trains that we've generated according to what we think these inputs are likely doing when the mouse is being presented with drifting grating. So this is a layer 2-3 pyramidal neuron in visual cortex, and it's supposed to simulate what a pyramidal neuron sees and does when the mouse is being shown a drifting grating. And so we have certain experimental data on how frequent those synapses are active. They have typically firing rates on the order of a few hertz or even less than 1 hertz. And importantly, they have different weights. So most of our synapses are quite weak, but there is a tail of strong synapses. It's a log normal distribution of synaptic weights. And that's what people generally find if they measure synaptic weights. And we also build in that this is not a random thing, but two neurons have typically a strong synapse between them and or are connected at all if they share some of their response properties. For example, if their responses to a certain drifting grating is highly correlated. So all of that we try to build in. That's the presynaptic side. We have like a presynaptic input model, all implemented in NEURON and using the NEURON spike generators and the ways of handling spike trains. And then there's a postsynaptic neuron constructed very much in the spirit of what Dieter just explained to you, how you have to go about this. This is hand-tuned, and it's supposed to contain or represent the excitability of soma and dendrites of a layer 2-3 pyramidal neuron to have enough sodium channels to support decremental exponential backpropagation, to support some kind of sodium spikes. We don't model calcium spikes, but we do have calcium channels. So if you were to kind of simulate a calcium imaging experiment, you could do it, but this cell does not make its own calcium spikes. That's something we omitted from the model. So as always, you have to make choices about what you include and where you stop, because you can't include all the details. And that's about it. So 2 components, the input model and the postsynaptic neuron. And here's a movie of what the spatiotemporal waveform looks like. It just ran across. I'm not going to restart it, I think. No, I am restarting it now. So you can see time moving as the vertical strip, and what's coded in color on the right is the most depolarized membrane potential at a certain distance from the soma. So it's like a projection over all the dendrites at a certain radius from the soma, and the color is determined by the dendrite that is most depolarized. And what you can therefore see is those orange and brown and yellow, basically dendritic spikes. And we have a detector for dendritic spikes. It's these red triangles and the white symbols. That is for sodium channels. So whenever there's a white symbol, a threshold sodium channel has been crossed to say that there's active sodium current going on in that dendrite at that time. And the first time it happens in a dendrite, it gets a red triangle. So it's the initiation of a sodium spike. And the orange triangles are for NMDA spikes. So when an NMDA spike has been initiated locally, it gets an orange triangle. So we have these detectors for the local spikes that we can use to account for how many synapses do we need to make a spike, and how many spikes do we get once the cell makes an exponential at the soma, which is the blue triangle, and the backpropagation indicated by those white symbols going almost straight up all the way. That's backpropagation of a somatic action potential. Yeah. Any questions about that while it's still running? Yes?
So it looks like the spikes are generated at different heights.
Yes.
From the same cell?
This is all from the cell. This is an excellent question. And as you can imagine, a suitable location to make a spike is one that has a high input impedance, where a single synapse or a few synapses can make a bigger difference than in a location with lower input impedance. Where do you have a high input impedance? Well, at the termination of a branch. Right? There is typically twice as high as in the middle of the branch. And so many of those little runs of white symbols that are initiation and propagation of a spike start at or near terminations of dendrites. And there's 2 kinds of dendrites in this cell. There's the basals, right, that tend to kind of stop at about 150 from the soma, and there's apicals that go up to 300 from the soma. And so what you can see kind of in the lower row, that is basically terminations of the basals and their dendritic spikes, and in the upper row, it's not as clear, it's the apicals. Okay, I think we should be able to, yes, great. So, now we're too far. So these are the questions we want to address using this exercise. We want to know what kind of synaptic inputs and via which postsynaptic mechanisms we get the somatic output of this neuron. So really kind of the initial big question mark, what is the rule this neuron uses to create output given its inputs? And you can also turn this around and ask, so which synaptic input patterns are especially strong and suited to produce output? And then how is it that this works this way? So how would you have to conceptualize the effect of the input via dendritic spiking to action potentials at the soma? And this is what I told you. We have 2 models, one of the presynaptic inputs and the postsynaptic excitability, and we can record, of course, from everywhere because it's a model, unlike in an experiment where we can record from only one location. But we can use experiments in hopefully a useful way. So here is the background to our distribution of synaptic weights. Is this paper by our colleagues, where they first showed anesthetized mice a series of still natural images in vivo and did calcium imaging to measure how similar or not 2 neurons respond to those images. And then they plot for all the pairs of neurons that they had in their data the response correlation. You can see most neurons show weak response correlation, so they respond differently. But there's some pairs of neurons that are really alike, that think alike, and they have a very high response correlation. It's the tail. And then independently of that, in panel D, you see once this is in a slice and you have identified which in vivo cell is which in the slice, and you patch those cells and measure EPSPs by making one spike and doing voltage current climb in the other, you can see that most of these have fairly small amplitudes, but there's again a tail of very strong EPSP amplitudes, strong synapses, the log normal distribution. And they're not the first to find this, but they're the first to link the two things. Because what happens is that if you have a high response correlation, so the cells like similar features in the visual world, then there's a much bigger chance of these connections between them to be really strong. Basically, it's a little subnetwork that is talking directly to each other and reinforcing each other about a particular feature that it likes in the visual world. And so we built this into our presynaptic input model. And the other thing is that from our own lab, we have lots of dendritic recordings in vivo from Spencer Smith and Ikuko Smith that tell us how the voltage transients in these cells look like when they are being shown a drifting grating at different orientations. And so here is a model response that really looks like what Spencer and Ikuko would measure in the experiment. So the black is the soma. You can see this cell is tuned. It prefers 90 degrees, so moving upwards. And it doesn't spike if the grating is moving horizontally. And so that's what one cell typically might do. And this is what a dendrite looks like at a distance where they could also patch. And so I'm going to challenge you now to tell me which of those is the model and which is the experiment. Any ideas? Votes. Okay, who thinks the model is pink? Who thinks the model is blue? I think the blues have it. So the trick is to see the scale bars. They are varying much more in the data, and that's an artifact of the experiment. If the series resistance is very large, then the voltages are much smaller than they would be in a perfect recording. When that's a recording artifact, not a biological change. But otherwise, from the waveforms, it looks very similar. I agree. So that's our other experimental hook, that we wanted to satisfy all the data from in vivo dendritic recordings. And now that we have tuned this model, we can play games with it. So for example, we can use our detectors for sodium spikes, the little red triangles, and we can now look back in time and in space. So at time 0 and at distance 0, there was a red triangle. There was a local sodium spike being initiated. And now we have, of course, our history of synapses that were active, and we can see where and when synapses were active that led to the sodium spike. And you can see the sodium spike is mostly, and this is kind of background subtracted, you can see kind of above 0 the extra synapses that happen by chance, because everything is random, over and above the average synaptic input rate. And you can see that they extend up to 50 or 80 microns from that location, and they go back in time, at the slight diagonal, a few milliseconds. So synapses can cooperate to make a sodium spike over a distance of, let's say, 50 milliseconds and a time of 2 micrometers and time of 2 milliseconds. But most of the synapses that drive it are fairly local. The big red blot is clipped. So most synapses are right down there. And for NMDA spikes, it's really mostly local synapses that drive it. So now knowing that these are the relevant synapses, we have defined this red window. And we are now going to count how many extra synapses are in those red windows here before a sodium spike or an NMDA spike. And so that's the result. So the colored bars show you it's about 2.3 synapses per sodium spike and about 1.6 per NMDA spike. And that's compared to what would happen in a random time window, so the same red rectangles, but at random times, not linked to a sodium or an NMDA spike. And you can see it's about, I would say, roughly 1 extra synapse that makes the difference. So it's really, really sparse. And you can see the histogram here. So if you now count how many synapses anywhere in the cell have happened extra to make this spike, then there's a fair chance that it's only 1 or 2. And really the majority of NMDA spikes is made by 1 extra synapse. And so now, of course, not shown in this slide, these are not average synapses. These tend to be the strong synapses. So if a synapse single-handedly, quote unquote, triggers an NMDA spike, it's of course not doing that. The reality is that there's a whole history of hundreds of synapses being active and keeping the cell depolarized by 20 millivolts above rest. So what Dieter talked about, right? You have this constant input in vivo that depolarizes the cell and makes it basically ready for the single synapse to have an effect and make a spike. If there was silence and the single spike were active, it would not make a spike at all. So single-handedly has to be taken with a big grain of salt. But it's the strong synapse that can do that. If you have multiple synapses contributing to, say, a sodium spike, many of them can be average synapses too. It's then the one out of maybe 5 that does the heavy lifting that is the strong one. And so to really show you that a single synapse can make the difference, I want to kind of show you this manipulation that we did. So we have a standard simulation where all the synapses are present. And notice the one with the star, so the one just above, just before the red triangle. We took that one out. So we basically have our spike train from all inputs, like frozen noise. We repeat it twice, but the second time we leave out exactly one synapse, and that makes the difference between this dendritic spike being initiated or not. So it's really an all or none thing, and you can demonstrate that for dendritic spikes. So I showed you the movie. Basically, what we think is happening is that this neuron does make local, at least excitability. It uses excitability. Whether this is really a spike that could be observed or initiated on its own is something we don't really know yet in experiment. In this model, certainly it looks like that. So we have dozens of spikes that keep depolarizing the cell up until it reaches threshold for sodium action potential and backpropagates into the dendrites. And so there is some support for this idea that this is real. And I'm going to switch here. This comes from experiments where people blocked NMDA receptors in vivo. So there's intracellular blockers for NMDA receptors called MK801. It's a drug that blocks the open channel. And if you put that in your patch pipette and you patch a single neuron in vivo while the mouse sees a drifting grating, this neuron will stop spiking. So that proves that NMDA receptors on all the nonlinear dendritic excitability, let's not call it necessarily spikes, that they contribute, is essential for producing any output. Without them, there's no output. Sodium channels are also important, but a bit less so. So we do know that there are very likely dendritic excitability in some form is always happening when a cell has been making an output spike at the soma. So here's now a similar story that we had for synapses before a local spike. Now it's local spikes before an AP at the soma. And you can see that NMDA spikes, being a bit longer lasting, they last 20 or 30 milliseconds, they can influence down to about minus 40 milliseconds whether there will be an action potential or not. Sodium spikes are a bit less long-lived, they are faster. But these are the kind of the hotspots here. They correspond to what Daniel asked about, which is the preferred location in this morphology where spikes are initiated, which is the terminations or near the terminations of the basals that corresponds to this hotspot, and the apical dendrites, which corresponds to this hotspot. And we can then count how many extra NMDA spikes or sodium spikes do we need per action potential, and the answer is a few. So up to 10, for example, or extra would be extra 6 compared to random times. So again, we talk about small numbers. It's discrete, it has kind of sparse feeling, it's a few synapses and a few spikes that make all the difference. So that's the answers that we think we have. We've shown that it's fairly easy with a model that has realistic amounts of synaptic input and realistic postsynaptic excitability, that the inputs expected during sensory processing when the animal is shown a strong visual stimulus, that these inputs can trigger NMDA and local sodium spikes, and they in turn can trigger action potentials. And it's a small number of strong synapses that plays an outsized role in triggering those local spikes and therefore also the somatic action potentials. So it is the local excitability in the dendrites that makes the neuron a stronger amplifier for the strong inputs. So fewer inputs compared to, say, a point neuron can create an output in this neuron. The circuit can live with a smaller ensemble of neurons that do all the processing, and all those other neurons freed up to do something else can do something else than process orientation of drifting gratings. And I'd like to finish by thanking Lea Götz, who's PhD was this project done in the lab of Michael Hauser, and the data and models that preceded this, they were provided by Spencer, Ikuko, and Tiago. And these neurons are by Hermann at the back, synthetic neurons. So if there's time left, I'd like to now switch to some presentation with the web browser. And I'm hoping that I'm allowed to exit. Say again? Yeah. I probably won't use all of it. So here, I've prepared some pages. Oops, I have to drag this over. So that's the neuron documentation. And I usually go here. And there's this thing called index. And so, for example, we can look at create. Where is it? Sorry, I have to probably switch to Hoke. Done it? Yeah. There. So here are examples. You can create multiple sections at the same time, separate by commas and so on. You can have arrays of sections. That's all you ever want to know about the create statement. Then it's alphabetic. The next is connect, how to connect up two sections, and so on. So this is the documentation that I find very useful. Then there is ModelDB. So by having, oops, where are we? Oh, I have to drag it over. By having this database of published models, we have a way of actually doing reproducible science. So anyone can download this and reproduce the published paper and use it as a starting point for their own work. And I'd also like to demo this. So exactly this model that I've downloaded already. And so here's the README. It explains how to run it. It has lots of information here. Here's an example mod file, for example, that you should see. This is a sodium channel. Lots of comments. And then this is the code that's being compiled and linked to the main executable of NEURON that allows you to use a private sodium channel model, which this model does. And so on. So this is the HOC code. We're going to run this code. This is creating the soma, accessing the soma, right? It's very much the same style of the simple example that you've seen before. And so here is how to run it. Where am I? Oops. So what we get is again the main menu coming up here, the run control window, and then we get an extra window that allows us to choose a cell type. It's just a list of buttons, and we're going to choose layer 5 pyramid. And this then brings up, has it done so? Where is it? Yeah, maybe we skip that. I can't see the necessary windows. So finally, we have neuromorpho.org. Which is searchable, so you can, for example, search for Purkinje cells. And I've done that. And I get this huge list of different labs and different species. So there's the species, and then there's Martone lab. Dendritica is kind of our own collection. And if I want to see that Purkinje cell, well, I did it just before, that's what it looks like. This Purkinje cell, and you can download the morphology file in a form that NEURON can read and use for simulations. So these are resources that are very useful for general work using NEURON. And there's much more stuff I didn't have a chance to talk about to do with interoperability with other simulators, with like meta simulators, where you can have a model description that can then be translated automatically to NEURON as well as other simulators. There's a whole ecosystem that NEURON is part of. Okay. I should maybe finally bring up the thank you slide, but I did it already. So thank you, and do you have any questions?
Thank you very much, Arnd. So we've got time for one or two quick questions. I'm checking on Zoom. I do not see anything on Zoom. Anybody in the audience? I mean, I guess this is one of those things that you just really need to practice. And
yeah.
Probably during the workshop, actually, you will have the chance to play with this and ask actual questions. There's one Dieter here, and then one there. Okay.
Of course, yes. So you can explicitly make a set of compartments for every spine, so like a spine neck and a spine head, and then if you have 100,000 spines in the Purkinje cell, then most of your CPU will do the spines, which is going to slow down your simulation considerably. So most of the time this is not being done, except for maybe the one or so spine of interest where a particular synapse sits that you want to study that has to be an explicit spine. But in general, what one does is one simulates an equivalent model where the extra membrane capacitance and resistance in the spine membrane is like folded into the shaft of the dendrite. So we take the same morphology, but we modify the effective surface area of the dendrite by multiplying membrane capacitance and membrane conductance. And then we have a model that behaves like a neuron with actual spines. So there's a little trick to simulate spines quickly. But yes, so in the model I presented, of course, we take into account the spines. I didn't mention it. It's an obvious thing. Everyone does it.
So I'm interested in the details of NMDA spikes versus sodium spikes. And are there specific types of synaptic inputs that each one decodes, different sources or different timings? And how many of the action potentials are ultimately contingent on one or the other, or even both?
Yeah. Good question. So let's see if I can get this thing back. Maybe let's go here first. Yeah. You can see from the way we detect NMDA spikes and sodium spikes that somehow with the sodium spikes we clearly captured not only the single local synapses that triggered it, but also the other synapses that also ran at a slightly earlier time and at some distance from that single synapse. And so we are very happy with that, and we think that that's plausible. That shows how in space and time synapses have to be staggered to really be effective at triggering a sodium spike. The detector for NMDA spikes had to be more tricky. So we did it based on the extra nonlinear current at our synapse of interest, which is why it's probably detecting only these things based on the one synapse and ignores what all the other synapses do. It's not as easy to have a clean detector for NMDA spikes, and we maybe haven't fully succeeded in building one. Anyway, so this one basically says NMDA spikes are triggered only by the local synapses. Other than that, for both types of spikes, what is true is that we know from the distribution of synaptic weights of those synapses that do contribute single-handedly is that these tend to be the stronger weights. I haven't included it here, but it's in the paper. It's the panel right next to these panels in the paper. You can look it up, compared to the overall distribution of synaptic weights. If, as I said, multiple synapses contribute, then the others can be average synapses, even weak ones.
But both of those events can remain local in their little dendritic branch, or they can make it all the way to the axon.
Well, most of them remain local. Basically, what happens is that there is one location where the spike detector is triggered first, and we assign that, say, the red triangle for sodium spikes. And then, like in these cases, you can see that it propagates to the end of that branch, and of course, there it dies out because the branch terminates, but it also propagates down to the soma, typically until the next major bifurcation, where there's an impedance mismatch that also terminates this spike. So you have a propagating local sodium spike that fills this entire branch where it originated, but does not invade any of the parent branches. That's the typical behavior. But you can see right before the backpropagation exponential, something kind of more complex happened. Right? It's almost like forward propagation to the soma and then backpropagation. So it's not always that these spikes in local branches remain isolated. At some point, they become cooperative.
So how many of the action potentials coming out of the axon were originally dependent or triggered by such local spikes, and how many of them came about otherwise?
All of them, and that's this. So if we block NMDA spikes, and we do that by making the NMDA receptors ohmic, so keeping them as they would be at -70, so all the magnesium unblock, all the action potentials depend on magnesium unblock. If we don't allow that, there's no action potential. And that corresponds to the MK801 experimental result. So we need NMDA receptors to have a functioning brain. We can't think and we'll be highly schizophrenic or whatnot if we didn't have them.
Okay, so now let's thank Arnd again, and it's time for coffee break. So Arnd, thank you very much.